Hello, my name is Arshad, and today I'm going to teach you about biochemistry. Basically, I'm going to give you a concept about biochemistry and the most important topic related to that. So basically, obviously, we have a massive production of living organisms around us in our environment. So whenever we are talking about a living organism, what does it mean? That there must be something continuously occurring in the body, or there must be some chemical reactions that are continuously occurring inside the world or inside the body. So here I'm talking about bio. So whenever you are talking about bio, it means you are talking about a living organism. It could be a human being, it could be an animal, it could be a microorganism, or it could be a plant, it could be anything. And whenever the word chemistry is coming, it means that there must be some chemical reactions that are continuously undergoing kind of chemical transfusions, basically. So whenever a word biochemistry combined become, it means that any chemical reaction that is occurring in a human body or in any animal body or in any living organism. Okay, as there are many examples, if you are puking, if you are getting food, if you are drinking water, if you are having a urination, if you are sweating, every single thing is basically a reaction of chemical things. These all are producing in case of chemical reactions. Okay, so today I'm gonna teach you a very easy concept, and that concept is basically the concept of enzyme. Okay, so whenever the word enzyme comes, it means that in means it is a basically is a Greek word, and it is it is formed by two words. En means en, enzyme means yeast. Whenever a word yeast become, it means that there must be kind of decomposition of bacteria or any kind of decomposition occurring in the process or occurring in any kind of substance that is living. Okay, so enzymes are basically the biocatalyst. Catalyst means they speed up the chemical reaction. Okay, so it speeds up the metabolic process, it speeds up the reactions, and it also breaks down complex protein molecules that could be hazardous for you, that can cause obesity, that can cause many serious health issues like a heartache, like diabetics, like many other problems. So basically enzymes are very useful as per they transfer a massive particles into very small particles. Okay, and definitely, for everything, there are continuous factors. Here they are talking about temperature, pH, and substrate concentration. We will discuss about it later. Thirdly, the most beneficial point for enzymes are that they are used as a catalyst, but they will remain unchanged throughout the process. This is the most golden thing, that they are doing their function, they are giving themselves up, but they are not changing they are not changing their chemical or their physical properties, basically. Okay, now, whenever you're talking about enzyme, you must know that there are three parts of enzyme. First one is the most important point, active site. As you can see, the word active means anything that is continuously in action. So basically, active site is a part of enzymes that is on the part where any chemical or where any particle, where any food which you are eating or anything you are just taking inside your body is just getting attached. So basically, I'm gonna tell you, this is basically an enzyme. As you can say, this is an enzyme. So this side is basically an active site where any substrate is gonna be attached. Okay, So, what is basically a substrate? Substrate is a particle. It is a particle at which enzymes act. Suppose that if you're eating a bread, so a bread is a substrate. A bread is a substrate. A water is a substrate. A peanut butter is a substrate. A sausage is a substrate that you are taking inside your body. And what is a word product means? We all know products means conversion or a final product after reactions. Definitely, when enzymes and substrate are just combining together, they are producing some kind of chemical reactions, and after that, a final product occurs. Okay, as I was discussing about the metabolic reactions, that enzymes are basically used to speed up the metabolic reactions. So, what does the word metabolism means? We have always listened about metabolism, that metabolism, whenever a person is feeling so low or is becoming a fat or an obese person, people used to say that their metabolism, or doctors used to say their metabolism is very low. Why? Because what does it mean that their normal functioning is very low? Usually they digest, or usually they kind of react to some things lately as compared to normal people. So basically, I'm talking about metabolism. Metabolism is basically a set of biochemical reaction in order to maintain life. Definitely, if your life is not maintained, if your food is not, if you're not taking food properly, if it is not coming out of your body easily, it means that your life is in a great danger. You become a patient of many things if your metabolism is not right. Obviously, if your metabolism is not right, it means you are creating a hell of mess inside your human body. You are having a huge health disaster inside your body. So basically, metabolism is a process or is a reaction which is used to maintain your life. Then it helps an organism to grow and reproduce. Basically, it helps an organism to grow and reproduce. Similar, easiest thing, that it allows a human being, or it allows any kind of animal to reproduce or to grow. If I'm talking about plant, there must be some metabolism in them. If I'm talking about myself, I'm a human being. There are metabolism inside my body. There are reactions going inside my body. Metabolism is not basically only the food I'm taking or the kind of thing I'm just eating. Metabolism includes every kind of reaction. If I'm not getting taller, it means there must be something in my metabolism due to which my hormones are not releasing properly. Okay, so the third one is the most important function, which means that they maintain the structure. Maintain the structure means, obviously, we all have listened in our normal life that if we aren't having a food properly, or if we are having a very high cholesterol food, it will directly affect our heart, and our heart hole will be closed, walls will be closed, will be clogged. What does it mean? That our metabolism is not proper, that the oozing out of the dirty material is not getting quick, due to which the dirt is still inside my body and I'm getting sick and sick day by day. So also, it shows a very responsive behavior. How is it so? If we all know that you cannot digest some foods, for example, you cannot digest a bird, or you cannot digest a steel, or you cannot digest a plastic. Whenever you're gonna take bite them in, you will puke out them, you will vomit out, you will vomit out them. Why? Because your metabolism is not accepting the kind of material you are putting inside of your body. So what does it mean? Basically, it means that it shows a responsive behavior. Now, we are having two simplest types of metabolism. The first type is catabolism, and the first type is catabolism, and the other type is anabolism. Okay, when I'm talking about catabolism, it means that larger molecules are synthesized. What does the word synthesize mean? Synthesize means ordering. Basically, what does word ordering means? That it whenever you are taking something inside your body, the larger molecules will be synthesized. They will be into order. They will set themselves into order. For example, if you are eating food, if you are eating a bread, there must be some kind of minerals that are so responsive for your body. So your body will store it. Catabolism allowed that thing that the minerals would be stored for your body which are necessary for you, and some minerals that are not that are not even important for you, they will start passing throughout your body. So what does it mean? That they have selected, they have ordered the minerals. And what does the word anabolism means? Anabolism means that the larger molecules are broken down into smaller particles. The larger molecules are basically broken down into smaller molecules. Why is it so? Definitely, you cannot digest a whole piece of bread inside through, you cannot pass out a whole piece of bread through your trachea, through your windpipe, or through your food pipe, basically. Sorry, not from your windpipe, basically from your food pipe. It is very difficult to pass a kind of a big bite from your body, from your food tube. So anabolism, basically, when you are digesting the food, when you pass out the bite from your pipe, the metabolic reaction, from the metabolic reaction, anabolism occurs and it transfers larger molecules into smaller. So basically, first of all, always remember that firstly anabolism occurs, it will break down the molecules, and after that catabolism occurs. It is on the number two, that after breaking down of the larger molecules, catabolism will select that which molecules are basically important for us. Okay, so now I'm going to teach you about the working. Whenever you are talking about working, it means that there must be some process that are going on. Basically, as you can see that here I have written E. What does the word E means? E means enzyme. And what does the word S means? S is substrate. Then whenever enzyme and substrate will come together, it will become ES complex. It will become a compact kind of material which is an ES complex, and then after, when enzymes have reacted, it will be transferred into product. As you can see, as I have told you, that enzyme being a catalyst will speed up the reaction, but it will remain unchanged. As you can see, the product have been formed, but the enzyme is still over there. Looking at the picture, this is basically the enzyme, this is basically the substrate, and they have combined together and they have made a complex, and the complex have been then transferred into enzyme and product. As you can see, enzyme is still there, but product have been also formed. So now the most easiest thing I'm going to tell you, as I have told you about the working, so there must be some mechanisms. There are two types of models that was showed for the mechanism, for the proper mechanism of enzyme action. Firstly, we are having a lock and key model. Whenever the term mechanism become, it means that the first mechanism we're going to tell you is about the key and lock model. Obviously, the word lock and key, what does it mean? It is specified. You cannot insert any kind of key inside your lock. You need the specific key which you have made for that lock. So Emil Fischer was a scientist. He was a German chemist. In 1894, he proposed a model. He named them as lock and key model. What does a lock? What does basically lock and key model said? According to the lock and key model, enzyme and a substrate have a specific shape and they are exactly fitted into one another. As I have shown you over here, as you can see, the shape of enzyme and substrate are exactly similar to each other, that they can easily be compacted, and they termed it as lock and key model. But later on, this concept was turned out to be a failure. Why? Because definitely sometimes the food you are taking in its shape could be an inappropriate. As you can say that whenever you are just ingesting or you're just digesting your food, your bites are in a different order. They are so in a in a very bad order basically. So it is very difficult as they are not having a proper shape. So this model was failed. According to this model, the active site, the site where reaction takes place, was rigid and it remained unchanged. It had a very specific shape, but this model was called out to be a failure. After Emil Fisher, we had an American biologist. His name was Daniel Koshland, and in 1911, Daniel Koshland produced a second mechanism model, and he named this model as induced fit model. According to this model, active site was not rigid, and the active site can change their shape according to the substrate. Substrate was the kind of food that you are taking inside your body, but enzyme is always present inside your body. So, enzyme, the active side of the enzyme is going to change their shape due to the substrate they are possessing. So, this model was turned out to be the corrected model as per you can see in the picture that we have an enzyme, then we have a substrate. As you can see that enzyme and substrate have an irregular shape, and enzyme and substrate have a compact bond. As you can see, it is showing the compact bond, which means that it has basically the active side has changed themselves to the shape the substrate was having. Okay, so obviously always remember, as there are many factors that also whenever we are talking about something particular, there are the factors that affect their working. As if I'm talking about myself or a human being, if we are talking, so whenever you are talking about human being, there are the effects of factor on us. For example, there is the effect of winter on us that we feel cold. There is the effect of summer on us that we feel so hot. And there is the effect of spring on us that we feel so pleasant because of the flowers and the spring rainy seasons, that they all are having effect on us. Similarly, whenever you are talking about whenever you are talking about um, factors, we are having three factors. The first factor I'm going to tell you, as I have told you that we are having the very first factor. The first factor was temperature. The second factor was substrate concentration, and the third was pH. Now, talking about the temperature, as you can see in the picture, this is a line of enzymes which are continuously acting on an optimum temperature. Whenever the word temperature comes and you have to tell about enzyme, always remember that we are having an optimum temperature. We are having an optimum temperature. Okay, optimum temperature means the exact amount or the exact amount of the Celsius or the centigrade where the enzymes will start working. So basically the room temperature is, it could be 27 or any you can say. So whenever the enzymes are under 27 degrees Celsius, they will continuously complete their work. But as soon as the temperature will rise, you will start your enzymes will start denaturing. We have a very good example that when we are sick, whenever we eat, as our body temperature is higher, our body temperature turned out to be very higher. As if the temperature is high, that means our enzyme is not working. So that's why whenever you are taking food, you start vomiting out and you start vomiting the food out. Okay. So whenever you're talking about temperature, there are three factors you need to know: that there is kinetic energy, there is temperature resistance, then there is global structure lost. We know that kinetic energy is an energy which provides a kind of movement to the thing. Okay, so whenever temperature is rises, heat energy also rises. Due to heat energy, when heat energy is rising, the kinetic energy will rise. When kinetic energy will rise, the molecules will start moving very, 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 very fastly. That it will start then it will start then it will start collapsing, and when it will start collapsing, there would be a time that then there would be a denature of enzyme. All enzymes will stop working, so you call them as denaturation of enzyme. Now the next point, the most important, is substrate concentration. Whenever you're talking about substrate concentration, it means that the exact amount of substrate you are having inside your body. Now, when you're talking about substrate concentration, you always remember this line, that this point is showing that the substrate is basically doing their work properly. But then a time became that it is constant. Why is it so? It is because when you have a maximum number, suppose it's just a supposition that you're having amount of 11 enzymes in your body, and you have eaten 17 kind of substrates, so all 11 active sites are occupied, so 6 are still substrates are present inside your body, due to which your concentration will be constant, because it is not reacting. There is no enzyme over there, so it is not reacting, right? When it is not reacting, it's mean, considering the example, we have seen the people who are obese, who are fat. Why? Because they are eating so much. As you can see, they are eating so much that their all sides are basically covered, and there is a hell amount of food present inside their body which is totally unbroken. They have the biggest particles inside their body, due to which they are obese. Okay, when substrate concentration increases, but as I'm telling you again and again, if the substrate concentration is on increasing, it will become constant, because there is no enzyme active site is available. Whenever substrate concentration increases and enzyme is constant, it will lead to saturation of enzymes, that there is no active site available. When there is no active site available, it means that it will create a problem, and you will start getting fat. Because I'm giving you an example regarding to this, and the most important and the last one is basically the pH. When you're talking the pH, it is as similar to as temperature, that for the working of pH you need an exact amount of temperature. Every pH is basically every chemical is working under the basic pH. Okay, everything has their own pH. If I'm going to tell you that pH 7, the 7 is basically the natural pH the water is even having, that you can basically which is not irritating. As you can, if you can see that in some areas of the world, there is water that is kind of acidic or is burny, that it is creating irritation to your body. Why is it so? Because their pH is different. Okay, and the body of your pH is basically 7. So always, then I'm giving you an example. Basically, there is a point low pH and high pH. Some enzymes are there which kind of work in a high pH, and some enzymes are there which work in a low pH. So we are having an enzyme, basically pepsin. Pepsin is an enzyme which works on low pH. It is a kind of chemical, or it is a kind of enzyme which is in your stomach. And there is another enzyme, you can call it a trypsin. Trypsin is basically enzyme which works on high pH, and they are present in large intestine. Okay, so basically, these are the three factors: temperature, pH, and substrate concentration. As per, if there is a similar, if there will be a little change in any kind of these factors, it will definitely affect your enzyme. If the temperature would be higher, there would be a denaturation of enzyme. If the substrate concentration would be higher, it will create a saturation, that the fat will start storing inside your body and you can become obese. And if the pH is not valid, then it can cause burn inside your bodies. Okay, so now the last thing I'm going to tell you is basically inhibitors. Inhibitors are basically the blocking side completely. What does it mean? It means that it is a kind of chemical, or it is a kind of thing that that completely or temporarily block your active site. An active site, what is an active site? Active site is basically the point where chemical reactions are occurring. Okay, so and inhibitors could be antibiotics, inhibitors could be cyanides or anything. Definitely, when you are ill and you are taking antibodies and you become fat. If you are having if you are having hormonal issues, so they give you antibodies, and when you're having a kind of antibodies, or if you are having a kind of steroids, cyanides inside your body, it will start blocking your metabolic activities, and it will start blocking your active sites, due to which your metabolism is poor and you can become sick. And there are two types basically. For one is reversible and the other is irreversible. Irreversible are basically the type of inhibitors that cannot be reversed. If it is on the active side, it can never be undone. But reversible, it can be undone by giving some kind of medicines or some kind of surgeries, you can say. Okay, and then lastly, I'm gonna tell you orally there are some uses of enzymes other than being a catalyst. Their uses are basically they are used in food industries, they are used in brewing industries, they are used in paper industry, they are used to make biological detergent. In food industry, as the name of enzyme is the breakdown of yeast, so basically, enzymes are used in bread, in making of breads, buns, then butter and white things like this, etc. Which is sorry, pizza. The yeast is basically using over there. In brewing industry, enzyme is used to break starch and proteins, and when the enzymes are break down starch and protein, then they are being fermented for further use. And enzymes are basically used to make paper. Why? Because it lowers the starch from the given part. Okay, and biological detergent, when you're talking about biological detergent, there are protease enzyme and amylase enzyme. Basically, protease enzymes are the type of enzymes, and amylase enzymes are basically the kind of enzyme that are used to remove stain from your clothes or from your dishes. Okay, have you ever noticed that ink or kind of an oil or anything is just stick on your body? When it is stick on your clothes, you need to remove it, and for this you need a very good detergent. And there are many type of detergents with every type of detergent are basically using enzymes so that it can wipe out uh, the marks on your dresses. What does it mean? So basically it can cleanse your breath. So basically the last enzymes are used, the most important is enzymes are used to cleanse your body. It helps you to maintain yourself to be a good and a healthy person. So this is for today. Hope you will get my point completely. Thank you so much. Have a good day. Bye.